So we're continuing our tour through the diversity of research fields that benefit from infrastructures. And I'm really very glad, honored that our first speaker for this session is Janice Thornton, who is from the Royal Society and a member of the EMBL. And she's a senior scientist at the moment in the Bioinformatics European Institute that she has directed for many years. And I would just say in one word that from my understanding, even though I'm not in your research field, you have really pioneered new bioinformatic tools to revisit the way we would study the structure of proteins. And because of this, you also were the key person that developed this infrastructure with all these acronyms, you know, ELIXIR. For those who are not the European members of the ESFRI, we have to learn all these infrastructure names. So ELIXIR, and if I'm correct, the title of your talk is The ELIXIR Data Infrastructure Empowers Research and Discovery in the Life Sciences. So the floor is yours.
So I'd like to say thank you to ESFRI and also the Academy here for the invitation to speak. It really is a great pleasure. And it's a pleasure because I'm very happy to celebrate 20 years of ELIXIR. It feels perhaps sometimes to me like 100 years because I was involved very close to the start with trying to establish one of the first infrastructures, ELIXIR. And for those who are, so the talk is going to be divided into two halves. The first part is to tell you about what ELIXIR is, how it's organized, etc. The second part is to give you just two examples of the way that it's already had an impact, I think, on society. And basically, ELIXIR is a pan-European infrastructure for data in the life sciences. And we know that the volume of life science data is really rapidly increasing. This plot just goes to 2021, and we have 62 petabytes of data. This is really large. We also know that the data are very diverse. We don't just have one source of data, the genome. We also have the proteome and the transcriptome and all different aspects of biology that we need to consider. The other thing is that the data that we collect comes from all over the world. Although this is a European infrastructure, the data is global. It is freely available to all. And you can see from this slide, which is a snapshot of EMBL EBI data provision, that the data are used all over the world. It's really amazing. We have 2 million hits a day on our website. I mean, this is a phenomenal usage of data. And just for a little bit of history, you can see ELIXIR's development. I was first aware of the infrastructure program in probably 2005, and we started developing. In 2006, we were added to the ESFRI roadmap. I ran the prep phase and the interim phase, but then Nicholas Blomberg, who you can see here, has taken over as director very successfully. And it's really been, I want to stress, although it was built on many other things, actually there was nothing there in a pan-European nature when we started this. So this is really, it takes time, but it's actually gone maybe even quicker than I thought it would, actually. So we have here what we think is a sustainable infrastructure for biological data. You can see all the ELIXIR member states who are present, and you can see that it's organized with a hub, but with nodes in the individual countries. So this is really important. Here we see the web page for the French node. That is a very active node and part. And our nodes really provide the expertise and the knowledge and the data that goes to feed to make ELIXIR a success. So what services, because this is all about service provision, do we actually provide? Well, we provide databases and data resources. We provide the ability to interoperate between those different data sources, and that's very important in biology, which is a very complex network of interactions. We provide tools, software and workflows, compute capabilities, training opportunities, and data management. And our European connections really build these separate nodes into a single infrastructure. And that ELIXIR commissions services from our nodes, and those help to connect and integrate and sustain these data resources. And they also improve collaboration across Europe. We've had lots of EU projects, very happily, Accelerate and Converge. And the nodes provide the data management expertise and infrastructure to deliver them. We're very much a key life science element of the European Open Science Cloud, and you can see some of the other projects that we've been involved in. Jan-Erik Paquet this morning mentioned about FAIR data, and within, or as part of ELIXIR, one of our nodes has created something called RDM Kit, and that allows, it supports FAIR by design. It allows you to design your projects to create FAIR, that's freely accessible, interoperable and reproducible data. We also provide bioinformatics tools, or rather the nodes provide the bioinformatics tools, and we provide a sort of a directory that allows you to find the tool that you need to do what you want to do. And we provide a lot of training courses. So each node provides training courses for their own country, but we share the resources for training. For those of you who've given lectures to teach undergraduates or graduate students, you know how much effort it is to create the material. And by sharing material, we make the whole process much more efficient. And in fact, over the years, we've had almost 1,500 training events. So the reach of ELIXIR is quite phenomenal. So now I want to go on to just 2 examples of the way that ELIXIR has already had an impact. And the way that, so there's the infrastructure, and that requires really good technical people that produce that. But it's also very important to link closely to the scientists who will use and help to create this infrastructure. And we do that by creating scientific communities. And you can see on this slide, I don't know if you can read it, but if I go quickly round, we have plant science, metabolomics, 3D bioinfo, proteomics, rare diseases, human copy number variation, human data, marine metagenomics, microbiology, intrinsically disordered proteins, Galaxy, which is a workflow system, and food and nutrition. So we have these scientific communities that have got together. These are scientists who've come together to create these communities. And they interact with and build on the platforms, the tools, the compute, the training, the interoperability, and the data. But I just want to give 2 examples to show that although this infrastructure is quite new in long term, actually it really has had an impact. And those 2 examples, one comes from human data, and one is about 3D bioinformatics, which is my particular specialization over many years. And of course, human data is special because it's sensitive. It requires secure access. And so within ELIXIR, we have developed the European Genome Phenome Archive, which is based at EMBL EBI. EMBL is an international organization, so it's funded internationally by our member states. And this European Genome Phenome Archive was established, and it allows people locally in different countries to store sensitive data that cannot be transferred across boundaries. And that's very important, that we don't, that we create an interoperable service, but we don't actually exchange the data across boundaries because it's sensitive. But the metadata are transferred to the central EGA and can then be searched to see where the data are that would be useful for a given research project. And this really came into its own in the COVID pandemic. And what happened, the Commission decided to launch the EU COVID-19 data platform, which is based at EBI, but you will see it has spread across the whole of Europe. So the idea was to collect viral data, as well as human data, and also some epidemiological data. And so we built a COVID-19 data platform, and we did that very quickly. And that was possible because we'd already invested in this infrastructure before. And within about 3 or 4 months, we were able to provide this COVID-19 data portal. This was supported by some of the emergency actions from the Commission, and also involves EOS Life. And you can see that data portal, which actually includes genome sequences, but it also includes other data relevant to COVID. And as of yesterday, I think it was, I looked, we have over 4 million viral sequences. And this is really quite critical to us understanding the disease and understanding the new mutations that occur. And what's key is that the tools that were developed at EMBL EBI have actually spread out to many other countries, and they have been implemented in these countries. And you can see Norway, Sweden, Finland, just going around, Poland, Turkey, Greece, etc., have their own instantiation of this platform where they can store the COVID virus data from their own citizens in their own countries. And they can also share that data because that isn't human data, it's basically public data. So that can be shared and interoperated. But it gives an individual country the ability to store and to track what's happening in that country. You can see it's also been adopted in Canada and Brazil. And so that's the first example where it's clearly had an impact over the last last year, really. The other area that I want to talk about is 3D bioinformatics and structural bioinformatics. So for those who aren't familiar with proteins, they are the most beautiful structures. They do everything in our bodies, basically. And you can see what they look like. This here is insulin, which absorbs, of course, glucose from the blood into fat, liver, and muscle cells. So the situation is that the 3D bioinformatics community is focused on these protein structures, the 3-dimensional structures. They're very large, called macromolecules. And we have different aspects to that, but I'll just talk about probably 2. The fact is that over the last 50 years, in fact, since I started, when I started my research in protein structures, we just had 14 protein structures. We now have over 170,000. And so this is very similar to the astronomy, where the new microscopes allow you to see things you couldn't see before. Now we can see these beautiful structures, and the structures are important because they work. That's how they do their function. Without the structure, they don't work at all. So we have all these structures, and in fact, these structures have been stored since the early 70s in a database called the Protein Data Bank, which is communal across the world. So there is one database with sites in 4 different countries. And so we have 170,000 different protein structures. And in fact, we have over 1,500 different COVID protein structures now. This has happened very quickly. Despite that, this seems fast to me, but compared with the accumulation of protein sequences, it's tiny. So UniProt is the database that stores all the protein sequences. And of those, we only have structures for 0.027% of these accessions. That is a tiny weeny bit. And so, inevitably, people have been trying to take these sequences and predict these three-dimensional structures. It's a very obvious and easy, this is why I got into this world, because as a physicist originally, I wanted to understand how these sequences in one dimension could create these beautiful structures in 3 dimensions. So people have been working on this since actually before I started in doing research. And what's happened within ELIXIR is that we have a 3D beacons program which brings all the predictions that people from around the world make for different proteins. Now you will see in that, so this is a platform that has been developed by EMBL, and you will also maybe be familiar with AlphaFold. So this was the AI deep learning method to predict protein structure from function. And 2 years ago now, there was a meeting which showed that this AlphaFold program was able to predict structures really accurately, more accurately than had ever been done before, using deep learning and artificial intelligence. And what was surprising, because this AlphaFold was developed by DeepMind, which is a company based in London, and it's part of Google, but it's very independent, actually. And they came to EMBL and said, well, will you help us make all of our protein structure predictions available? And of course, we'd already got the tools to allow us to do that. So actually, they had funded the creation of the AlphaFold structure database. This is completely open access. So anybody around the world can access this data. You can bulk download it, etc. They started out by having 11 sequences, but recently they've added another chunk, and in particular I would mention the neglected tropical diseases and the organisms responsible for antimicrobial resistance. So they have produced predictions of the structures for all the proteins in these organisms for everybody to have access to and to use. And since we created this database, there have been almost more than 400,000 users since the launch. So this has clearly had a big impact on the world, if you like. And one example, which was from EMBL, is using this data, combining it with experimental data to build some complex, very large complexes. And what you can see here in the colored ring is the nuclear pore. It's the pore that allows stuff to go from the cytoplasm into the nucleus, basically. And this was built using these models obtained from AlphaFold, combined with some new experimental data. So this is real step forward. And of course, with all of these structures are really good when it comes to designing novel therapies, novel drugs. Without that, almost every drug design, almost every drug design process involves using protein structures and computational chemistry and docking, etc. And so they're one of the doing it by itself, sorry. One of the activities is to utilize all of this data, including the predictions, to design for drug design. And one aspect of 3D bioinfo is to do that. Right, so I'll go to the future. It's obviously keen to get on with it now. So what's going to happen in the future? So there are many national genome projects around the world. There's one somewhere in there is France, but there are also many others. And basically, in total, when I made this slide, there were about 220 genome initiatives worldwide. So as a consequence, we are going to get, we've got a lot of data already, we're going to get much more data. It's also true that there are many new technologies coming along in the life sciences. In particular, the single-cell genomics, that's when you can take a single cell and look not only at the genome, but the proteome, the proteins in that cell. So we have about 200 different cell types, and you can look at each cell type and at individual cells to see what's going on in them. It's amazing. You've got the metabolomic modeling, that's looking at small molecules. The multi-scale modeling, the whole linking genotype with phenotype. So it's clear that will generate more data. And also, there's all the applications of these data in, if you like, in society. And here I just list the bioenergy, the biodiversity, soils, cancer, and human variation. Much, much more data still. So it's very clear that we've been living through a genomic revolution, but we will live through another revolution as the impact of data science and our understanding of life sciences are applied to medicine, biotech, and the environment. So if I'm allowed just one comment before I finish, this whole project has really been about bringing all the different scientists around Europe together. Now, you're all aware, of course, of Brexit and the Swiss situation. That means, at the moment, we aren't associated with Horizon 2020. This is absolutely, the UK government promised that we would be, but it's stalled at the moment because of political things. So it feels as if we're being used a little bit as a bargaining chip. And one thing that you could do, if you value having the UK and having Switzerland as part of these collaborations, and we've seen in all the slides, there's clearly a presence from those two countries along with the rest of Europe. And there is something called Stick to Science, which is about Europeans potentially saying that it's important to them that the UK and Switzerland remain involved in these pan-European projects. As I said, having devoted 10 years of my life to setting up ELIXIR, I am totally mortified, as are all the other scientists that I know in the UK. So if you want to try and help, perhaps if you go to the Stick to Science website, you can sign your name to show that you support this going forward. So thank you very much.
Okay, we can take 1, 2 questions, please. Yep.
Thank you for your talk. You mentioned AlphaFold, and presumably among the lineage-specific proteins for which there's no evident sequence similarity, there are some novel folds that have been predicted. Has there been a coordinated effort to try and crystallize or otherwise determine the structure of those proteins, and do we know how good AlphaFold is when there isn't a clue to help it?
Yeah, it's very clear that there are some structures that are novel, completely novel. There's a wonderful one that's called the heart protein that looks like this. I mean, it is beautiful, but at the moment, there isn't, to my knowledge, a coordinated effort. However, structural biologists are very keen on finding new folds, so I'm absolutely convinced that there will be. But actually, it's very difficult because, as you know, families diverge in all sorts of ways, and it's actually quite difficult to be certain whether something's a new fold or an old fold that's changed. So I think that will come. And it's clear with the usage of the AlphaFold database. I mean, I've been amazed how much it's been used. And it's really reinvigorated our structural biology colleagues to go ahead and determine lots of structures. And some of them are beautiful. I mean, they're three-layer structures that haven't been seen before. So, but how good they are, still to be determined.
Another question? If not, thank you so much. And now we're switching to linguistics, something which is related to the CLARIN infrastructure through the first talk of a young researcher here, who is, and I'm gonna, sorry for spelling badly your name, Kaja Dobrovoljc, more or less. And you will tell us about open language resources for smarter artificial intelligence. It's yours.
Actually, Dobrovoljc in Slovenian means the one who is always in a good mood, who's happy, so which is quite accurate. I'm very happy to be here. Let me just see how the slides work. Okay, very happy to be here, to be given this opportunity to say a few words on language resources and why they matter to artificial intelligence. Not the correct button, I see. Ah, it's the green one, naturally. So, a few words about me. I'm a linguist by training. I work as a postdoc researcher at University of Ljubljana and the JSI Institute, where most of my work has been focused on language resources and technologies for Slovenian, which means that my work, my research work, is closely associated to the CLARIN research infrastructure that we are also celebrating today. So what is CLARIN? It stands for Common Language Resources and Technology Infrastructure. It has been a part of the ESFRI roadmap ever since 2006, and it has the ERIC status since 2012, and has also been recognized as one of the landmark infrastructures. The basic idea behind CLARIN is that it provides easy and sustainable access to digital language data in various forms, mostly intended for scholars in humanities and social sciences, but also to other interdisciplinary researchers working with language data. CLARIN does so by also offering advanced tools that enable easy discovery and exploitation of these resources wherever they are located. And it does so through a single sign-on environment. In addition, it's also a hub for knowledge sharing in the field and is one of the research structures belonging to the social sciences and humanities cluster. So in terms of organization, CLARIN works as a distributed network with 70 centers across Europe and 22 full CLARIN members, one of which is also the Slovenian National Consortium, CLARIN SI, which has been active since 2014 and basically deals with language resources, services, and tools for Slovenian, but also for other South Slavic languages in the region. But of course, I wasn't invited here to tell you about the big picture, the infrastructure, but rather to illustrate how this particular infrastructure has had a very important impact on my personal research work. So let's get back to language technology, which is a branch of artificial intelligence that enables machines to understand human language. I wouldn't have to prepare this slide to begin with because we saw the perfect example of language technology in the beginning when the introductory talks were automatically translated from French to English, right? So a perfect example. But of course, language technologies are everywhere, from search engines to voice assistants to tools for grammar correction, for sentiment analysis, and so on. So different everyday applications that need to make sense of the language we use. But how do computers learn a language? Well, it's actually very similar to how as humans go about learning a new language. We try to read as many books as possible in that language. We listen to the radio. We throw our money into the dictionaries and grammars we never actually use. And we try to have as many interactions in that language as possible. And it's the same way with computers. We feed them all these different types of language data, such as corpora, lexical resources, and other types of language resources that the machines then use to learn from by using different machine learning techniques to produce their model of the language, so their statistical understanding of how the language works, which you can imagine must be done for every single language. So imagine the number of language resources that are being developed and used in Europe alone. For Slovenian in particular, most of my work has been focused on a very particular type of resources called tree banks, which are still one of the most popular ways to describe a language's grammar in a machine-readable way. So what exactly are tree banks? Well, simply put, they're collections of grammatically analyzed sentences in the form of a tree, which, let me give you an example, looks like this. So if we have a sentence such as this one, France will host the ESFRI 20th anniversary conference, a tree bank would also provide information on the morphology and the syntactic analysis of this sentence. So each particular word in the text gets different kinds of linguistic labels assigned. For example, telling us that France is a proper noun in singular number and that it acts as a subject to the verb host. So with a little bit of imagination, you can imagine this forming a tree-like graph. Of course, this is a very time-consuming, meticulous linguistic work, but it has been proven to be very useful, not only for linguistics to develop data-supported linguistic theories, but also for language technologies to develop the so-called parsing tools that learn from this data, so this manually annotated data, and are then able to produce the same kind of grammatical analysis, the same kind of annotations on much, much larger collections of texts, which in turn is very beneficial to anyone doing research on that data because it enables them to do much more complex data analysis than looking at words alone. So how did we develop a tree bank for Slovenian? Well, it's been a very long process that has heavily depended on CLARIN maintained data services and tools coming from all over Europe. So we first started out by taking a pre-existing data set, pre-existing corpus from the CLARIN SI repository. We manually annotated it using a tool that was developed by the German CLARIN Center, and the resulting tree bank was then published as part of a much, much larger collection of tree banks, which is hosted by the Czech Lindat Klaraja repository. And the Slovenian tree bank in particular has also then been integrated into the local services provided by CLARIN SI, which enable now linguists to browse this data and make different kinds of research on top of it. But perhaps the most significant impact this tree bank, this data set has had is in the field of language technology. Because ever since its open source publication in 2015, it's been used in modeling several state-of-the-art parsing tools coming from all over the world, including a tool that's being developed locally by the CLARIN Knowledge Center for South Slavic Languages, and which is actually one of the best performing tools for grammatical analysis of Slovenian. So I put some numbers here to give you a better perspective. So this is the numbers you see are actually, it's the tool's performance. So the percentage of words that the tool gets right. So for example, for the full morphological analysis, so saying that something is a verb of this gender and number and case, the tool makes less than 3 mistakes in 100 out of 100 words analyzed, which I think we can all agree is quite a remarkable achievement, especially if we consider the complexity of the task, so this kind of grammatical analysis as we've seen before, but also the speed with which it does so, right? So I think this is a perfect example of a very tiny, yet very, very smart piece of artificial intelligence that wouldn't have been possible without the language resource in the background, but also the infrastructure that was needed to develop it, right? So to conclude, I'd like to emphasize once again that open language resources are essential to scientific advances in all language-related disciplines, including artificial intelligence, and that the development, distribution, and exploitation of these resources has really been made significantly easier by the ESFRI CLARIN infrastructure. And we've seen this is definitely the case for Slovenian language. And to end on a more humanist note, I think it's also important to point out that it's not just about the infrastructure, but also the people behind it, which is especially important for early stage researchers that need some support in navigating this complex infrastructure landscape. So thanks again, CLARIN. Thanks again for your attention.
One burning question? If not, thanks a lot. And now we are turning to biology with Maria Ina Arnone, that comes from the Stazione Zoologica di Napoli. She is connected to the infrastructure on the European Marine Biological Resource Center, EMBRC, and she will tell us about what determines the form and evolution of a living organism, lessons from the sea. It's yours.
Thank you very much for the introduction, and I want to thank again the Academy of Science for giving me the opportunity to talk about my work and to talk about EMBRC in this wonderful meeting. So here is an outline of, this one. An outline of the story I will tell you today. I will give you first an introduction about Stazione Zoologica, Anton Dohrn, and the EMBRC infrastructure. Then I would like you to introduce to the type of work we are doing in my lab, particularly using the sea urchin embryo and the approach of studying gene regulatory networks in evolution and development. And then I will give you first a insight into a novel technology that open many new avenues in this field, and use just one example to show you this novel integration of approaches that we are using in our lab, for which infrastructure, European infrastructure, are fundamental. So first of all, let's talk about the sea. So life, as you all know, began at the sea. And it was more than 500 million years ago that the major body plan of the animals that we see on Earth nowadays were already established. And this is clearly demonstrated, was discovered actually in 1909 when the Burger Pass in British Columbia was discovered, and these fantastic fossils dating 500 million years ago were discovered. So not only the sea has been the cradle of evolution, but the sea has been and continues to be nowadays an incredible source of experimental organisms for which you can do research and do important discovery for life science. This was a very clear message, was a very clear thing that for Anton Dohrn, German scientist, who decided to fund a new institute in Naples, the Naples Zoologica Station, with the idea, with the aim of proving that Darwin theory were true. And actually, there is an endorsement, a letter of Darwin that addressed to Anton Dohrn in response to a letter in which we have to say Dohrn was asking for money, okay, for support for this initiative. Darwin didn't have money for that, but at least give a series of books for the library, and this great endorsement by saying, I fully believe that you have done a great service to science. And this was true, this was important, but actually the dream of Anton Dohrn was even bigger. He had a vision about what a zoological station is. Think of the name station. Why use the name station? He invented this name. Because the idea of Anton Dohrn, the vision of Anton Dohrn was of making a network of marine stations. At that time, it was the only research facility that you could imagine where you could have live organisms to study. And the idea was that researchers were allowed to do their research, work hassle-free, and then move from one station to another, like the station, like the train station. Okay? So Anton Dohrn did a great work, and as a manager of science, he even funded an aquarium to fund research, and so on. But his dream was limited to the Naples station, although in the other station in Europe were starting in the same year on this model, some even on the structure following the model, like the Plymouth station. But this dream of Anton Dohrn actually came true through the ESFRI infrastructure of the European Marine Biological Research Center, which has the aim of allowing fundamental and applied marine science. And that's what I will tell you about today, a little bit of the research that you can do with this type of infrastructure. And in this map of Europe, you can see which are the countries that are now involved in EMBRC. And as you can see, France is now coordinating, and the headquarters are actually here in Paris. And it was in the preparatory phase that actually Stazione Zoologica had a key role in EMBRC, in funding EMBRC. And I want to go back a little bit, a moment to Marine Station, Stazione Zoologica Anton Dohrn, and to that period after the foundation, that is at the end of 1800 and beginning of 1900, when many scientists from all over the world were gathering in Naples and made some fundamental discoveries using marine organisms, discoveries that paved the way to what is modern biology. And I want to give just an example to introduce then the model system I use nowadays. That is this famous discovery made by Boveri at Stazione Zoologica, in which he was using the sperming eggs, so eggs that were fertilized by 2 sperm instead of 1, and was studying the fate of the blastomer that were dissociated after this fertilization. And then he demonstrated through a series of very elegant experiments, I don't have the time to illustrate, but very, very elegant if you think that at that time only the word chromosome meant something like the name says, that is a body that can be colored. Nobody knew what was inside this body, and actually it was thought that they were all copy of the same thing. Indeed, Boveri understood that these little chromosomes, they were different one another, and actually that only the blastomer that was inheriting a complete set of these chromosomes could give rise to a normal animal. So to use the word of Boveri, only a precise combination of chromosomes represent the complete nature of the form of a living organism. You have to consider at that time was even the question, what is making the form of an organism? And actually, this is still the question that nowadays we are asking. What determines the form and ultimately the evolution of a living organism? Here you see two fertilized eggs. Okay, they look very much alike. Indeed, one will give rise to that animal, and another one gives rise to the other animal. How is that? What is the answer to this question? We know that the answer is written here in this huge book, that is the genome of each animal, that is constituted by only 4 letters. And how to read this book? We know quite well nowadays, thanks to a lot of bioinformatics, we know how to interpret what are the parts that here I colored with this different color bar that represent the genes, and that will be transcribed and then translated into proteins that then make the beautiful structure that we were seeing before. And we know that these proteins are ultimately what are making the difference between cells. So we know that during development, ultimately what happens is that some cells are fated to, let's say, make the air, and then they have to produce keratin. Some cells are fated to become the eye, and then they produce opsin, crystalline protein, okay? But what tells to the cells during development? Transcribe and translate that protein and not the other one, and in this precise moment. Everything is written in the same book. Everything is written in between these lines. But we will have learned in the last 10 years a lot also how to read through the line. But ultimately, the real program only experimentally can be tested by the reconstruction of what is called gene regulatory networks, that are this very complex structure that I cannot tell you about, but basically are an attempt to reconstruct all these commands that are written into the genome. Okay? And to do this, you can do a lot of prediction bioinformatically, but still you need experimental biology to first validate, but also to interpret what is written in there. And this is what we are doing in my lab by using marine organisms mainly, and we basically trying to understand how an organism is made, how an organ is made, or how a specific cell is made. We reconstruct the gene regulatory network that dictate that structure, and to study the evolution, we compare in different animals the gene regulatory networks that is at the base of the same structure in the different animals. So which are the animals that we use mainly? We use the sea urchin. And you see the adult here, that is this beautiful tube fit, but the main target of our study and of the story I will tell you today is indeed the sea urchin embryo, a larva. That is that tiny little thing that, that you see, well, that I'll show you better in the next slide, because otherwise I don't have a pointer here to especially indicate. So this little larva is less than 1/10 of a millimeter. So it's a microscopic object that live in the ocean. Is actually quite transparent. Here you can see a scanning electron microscopy and a micro CT that is a tomography, so X-ray, that show the structure of this transparent thing. You can appreciate still, and this is an optical microscopy at the end, and you can appreciate actually that there is a tripartite structure, a gut, a complete gut that allow for digestion of the food. And overall, this little microscopic animal that has 500 cells, so less than 2,000 cells in total, has differentiated cells for all the type of structure that are necessary to digest the food, to orient, to see the light, to perceive the depth, and to live in the ocean. And then to carry on what is the new program of the adult that will, after metamorphosis, derive from this little animal. So we have also developed large genomic resources since 2016, when the first genome, the one of the purple sea urchin from California, that is the main object study in my lab, but there is also the Mediterranean sea urchin that is coming along with. And we also recently developed what is so-called epigenomic and regulomics type of resources that helps in predicting what is this program of development I was telling you about. And this also are a little summary of the type of project, but this is the 2 minutes that are left. Oh, Jesus. So I will go very quick to this novel approach. I didn't realize that, I thought that, to the novel approach using the lab to study gene regulatory networks and development of animals. That is the single-cell transmitomics. That is a tool that allows you to sequence all the cells and then to bioinformatically cluster them to understand the different cell type. And why is not going on? And this type of graph, the different colors represent the different cluster of cells, and by validation of this, you can figure out which cells is representing what. And I want to tell you only a little bit something about the pancreas, the pancreatic cell type that we identified in this animal. You know that the pancreas is an organ that exists as such only in vertebrates. But in invertebrates, like the sea urchin, you can find some cell type that produce the same gene that are necessary to produce pancreatic cell type. And in particular, these exocrine pancreatic cells that were demonstrated here, we could also reconstruct the gene regulatory network and find that is extremely conserved with the gene regulatory networks of the vertebrates. And more interestingly, we also studied the structure of this cell by using, again here, an infrastructure and using a special scanning electron microscope that allows to detect and to reconstruct the cell of an entire organism, and making so an atlas of an entire organism. And we could find these special cells that are vesicles that, like the acinar cells, are oriented towards the lumen where all the enzymes are secreted. So in conclusion, with this type of approach, we could demonstrate that the long-standing theory of neurons, of pancreatic cell deriving by neurons, is actually true, and probably this event of co-option occurred much earlier than vertebrates, and it was present also in sea urchin. So sea urchin by humans diverged 500 million years ago. And in general, actually this, I was very happy to discover that the French press was interested in this discovery made in my lab, of the fact that the pancreatic cells of the sea urchin are so similar to the one of vertebrate. And the final message I want to give of the conclusion of this type of study is that with this example of the pancreatic cell type, we can see that actually an integration of atlases of gene expression, atlases of 3D structure, and gene regulatory network functional analysis in the context of an entire organism is actually possible, but it is possible thanks to this infrastructure. And this is just to thank the people of my lab and you all and the funding. And if I can give you just 1 minute more, I want to thank, I want to take the occasion here to remember a person who disappeared this year, Professor Mike Thorndike, that has been a great colleague and for many years, but has been a person very instrumental to the infrastructure of Assemble Marine first, and then EMBRC in the preparatory phase. And thank you all for your attention. Oh, so this was for the 15 minutes, the time. Okay, I rushed.
So we have time for some more questions.
Because I was rushing as crazy when I realized that the time was over. I didn't realize that was the 15 minutes time. That's why.
Oh, sorry, I was not on the microphone, sorry. So I'm asking again, I mean, who would like to ask a question? Is anyone prepared to? Yep.
Hi, that was sea urchins are beautiful.
I agree.
So you'll be aware of plans to sample the ocean round the coast of Europe with an EMBL and looking at that. What sort of things do you think would come out of that? Because that's looking at many different organisms potentially. So how do you think it will impact your very detailed molecular work when we have this knowledge of all these protists and things that live around Europe's coasts?
Sure. Thank you for actually giving me the opportunity. You are talking about planetary biology, right? Yes. And as you probably imagined from the names I was mentioning, I did all this single-cell transcriptomics in collaboration with Ethel Arendt, this coordinating project. We are all very excited about this large project that has been launched well, actually been launched last year, but was only this year that the funding arrived, and so this will be a great opportunity. And following what was similarly to what was done with the Tara collection all around the world, now a big campaign of sampling throughout Europe and Mediterranean Sea will be performed by sampling at the same time on the sea and on the coastline. And of course, we have a lot of places in Italy, and Naples is one of those which we are doing. And we are expecting to find many, many answers to our question. For example, if we go to the little sea urchin I was showing you before, we are studying a cell type that is in the apical organ of this animal, let's say the little brain of this larva. Are two cells that contains a photoreceptor, and that are responsible for, this is what we have recently demonstrated, for the ability of this larva to, depending on the circadian rhythm, to understand if how much amount of food is around, and if the food is scarce, the larva develop in longer arms so that have more cilia that can attract more food, okay? So we know that this larva does this in the lab. We never seen this in the ocean. We never seen how during the night and day cycle, actually these larvae are swimming up and down. These are all questions that with this sampling all around the world we can, all around the Mediterranean we can answer. But I mean, the amount of data that will be derived of that, you are well aware, will be huge. And actually, artificial intelligence is also acting in this a lot. The 3D structure of the cells I was showing you, that was done after the acquisition of all the scanning of this little larva. And actually, there is something I wanted to say. Today, was mentioned this fact of the remote access. The experiment of acquiring, of actually fixing the larva that was then segmented at the EMBL was done just 1 week before the lockdown started in 2020. We basically escaped from Heidelberg, okay? We didn't know if we could enter in Italy because it was at the beginning of the lockdown. Then the acquisition was started in the lab in Heidelberg, but thanks to the technology of nowadays, we could connect from our computer in Naples and see online, the acquisition going on, the acquisition takes 3 weeks, 3 weeks of continuous. It's a fantastic new machine that we were guinea pig and we used for free at the EMBL because it was just acquired. Actually, it was still in demo. It's the same machine as an electron microscope and a microtome. So basically, you have your sample, and then you start cutting and take pictures. Stacking and take pictures. Pictures means picture at the electron microscope. And you know, this is very costly in time and also in space. So we could connect to machine and see, for example, in this area, there is nothing. So don't take picture, go to this area. And we could command the machine in remote from Naples. But then you have to reconstruct the 3D structure. So this was done with the program, Amira is called. A program that was done by a student that did the master thesis on that and reconstruct this beautiful structure. And it took months and was done manually. Nowadays, we are using artificial intelligence to recognize specifically structure and so to do this 3D reconstruction. So yes, the future is there.
Okay, thank you for your answer to this question. So our next speaker will give a young investigator speaker. He's Alexander Schumacher from the Max Planck Institute for Social Law and Social Policy. His work is connected to one of the infrastructure which is called SHARE for the Survey of Health, Aging and Retirement in Europe. And he will tell us about determinants of the SARS-CoV-2 vaccinations in the population over 50, which is of interest directly to some of us, certainly.
So thank you for that introduction. This project is carried out by my colleagues Michael Bergmann, Tessa Virginia Hannemann, Arne Bethmann, and myself. And it came to fruition during the late months of summer in 2021 when it became clear that the vaccinations campaign were not stalling due to issues with distribution, but rather with issues of acceptance of the vaccine. And so we wanted to know who are these people that do not want to be vaccinated. But before we get into that, I want to say a few words about SHARE and how we, as a study that deals with the risk cube of the pandemic, dealt with COVID. So what is SHARE? SHARE is the Survey of Health, Aging and Retirement in Europe. It's a panel survey studying the life of Europeans aged 50 and older. And it has the aim of investigating the impact of policies among people aged 50 and older. There are 3 advantages of SHARE as a survey study. Oh, that was Shirley, excuse me. One, it takes into account the entire life course. So we have questions about the upbringing of our respondents, about education, about employment history, and even about the final stages of life. Secondly, it is a panel survey study, meaning we interview our respondents not just once, but repeatedly every 2 years, and so can track changes over time in their life. And thirdly, it's a cross-country comparison that covers almost the entire EU. And all those factors together allow for the causal analysis of policies in European countries. So what are the facts so far? We have conducted 10 panel waves since 2004, 8 personal interviews and 2 telephone interviews. And we are currently working on the fieldwork of the 9th personal wave. So far, we have collected 530,000 interviews with 140,000 respondents in 28 countries. And of course, that kind of coverage would not have been possible without considerable support from the European Union. Now, of course, like for anybody else, COVID changed a lot for our survey, and we had to interrupt the data collection for our 8th wave to protect the health of our respondents and our interviewers. We had to break off the data collection in March 2020. However, we quickly came up with a new plan, and only 3 months later, we resumed interviewing by telephone with a special focus on the COVID pandemic in July 2020, so only 3 months later. And we did a follow-up survey about 1 year later. These 2 surveys cover the most important life domains, as well as specific questions about vaccinations, infections, and pandemic-related topics like health and mental health, healthcare, economic situation, and social networks, all of which were affected by the pandemic, of course. Now, let's go on with an example of how these data can be used. Our goal was to identify who are the people who were hesitant about getting a vaccination. And we wanted to give a descriptive overview on the most important variables related to hesitancy to be vaccinated against COVID-19. The areas we looked at were demographics, health, and socioeconomics, but today I will be focusing on demographics and socioeconomics. The data we used is mainly from the Share Corona Survey 2, which included questions on vaccination status and vaccination intent, plus more individual information on our respondents. This is a great advantage of our survey data over administrative data. We can actually add further info to the pictures, such as education or household income, which stem from our regular share waves and the first Corona survey. So let's get into the results. Of course, as a cross-country study, we first have to look at country differences. On the vertical axis, you can see our 28 membership countries, and on the horizontal axis, you see the share of respondents who were already vaccinated against COVID. In yellow, you can see those who want to be vaccinated. In orange, you can see those who are still undecided, and in red, you can see those who don't want to be vaccinated. Now, keep in mind, these data are from the summer of 2021, so they might differ from the national vaccination rates today. But on average, at that time, we saw a vaccination rate of about 82% among the 50-plus population, and that actually matches quite closely the rates in administrative data at that time. And we can see quite significant variation across countries. For example, in Romania and Bulgaria, we are just about 20% of people who are already vaccinated, while in countries like Spain, Denmark, Malta, we are at about 95% of people who are vaccinated. But what you also can see in this picture is that to some extent where these differences come from, they seem much more driven by people who are undecided or not willing to be vaccinated than actually by the issues of distribution, so people who want to be vaccinated but are still waiting for their vaccination. So now let's find out some more details about these people who are hesitant. First, to demographic groups. On the horizontal axis, you see the share of respondents who were undecided or unwilling to be vaccinated. On the vertical axis, different groups of respondents. As you can see, younger respondents were more frequently undecided or critical of being vaccinated, as well as women were. Here we see that women were less likely to be vaccinated and less willing. And finally, the highly educated had a higher probability of being vaccinated, so they seem less redundant against vaccinations. Let's look at some important socioeconomic groupings. Again, the share of hesitant respondents on the horizontal axis. Firstly, looking at housing environment, you can see that respondents from rural environments are more frequently hesitant about vaccinations than respondents from cities. But really the most important differences can be found in relation to financial distress. Our respondents who were not able to make ends meet financially at the end of the month had a higher probability being refusing the vaccine than those who had no difficulties making ends meet. And finally, the same we can see for the risk of poverty. Respondents who were not at risk of poverty were much more likely not to be hesitant. The last variable I want to point your attention to is those respondents who were retired or employed. And here we see that self-employed or unemployed respondents were much more likely to be hesitant about vaccinations. So to summarize, we could identify some groups that seem not to have been reached by the vaccination campaigns as much as others. For example, people in financial distress or with a lower level of education, which could be quite useful for future vaccination campaigns. These were just some rough descriptors, but I hope you could give you some first ideas of who the unvaccinated among the 50+ population are. If you want to know more about this topic, we have a working paper published. Our current study is also under review in a peer-reviewed journal. But all that's left for me now to do is thank you for your attention.
Is there one burning question? Somebody who feels concerned because of an age issue, for instance? No? Everything's fine? Okay, people are educated here, so it seems that they are not worried too much. Thank you. Okay, so our next speaker is Pearl Dijkstra from Erasmus University in Rotterdam. Her work is connected to the infrastructure Odyssey. I think that you run it, right? It's called Open Data Infrastructure for Social Science and Economic Innovations. And the title of your talk is Science for Policy Requires Research Infrastructures for Policy. Serious.
Thank you for inviting me to speak at this birthday conference. As I was introduced, yes, I'm the scientific director of a social science infrastructure in the Netherlands, Odyssey. And it's a privilege to provide you with some background on how a data infrastructure like Odyssey enables policy-relevant research. But I'm also going to be talking about challenges that remain. Sometimes science and policy have fundamentally different emphases, and let me illustrate. First, there's the issue of specification. I'm going to take you to the top left-hand corner. In policy, there's a need to reduce type 2 errors, not flagging a condition. Whilst in science, the focus lies more strongly on reducing type 1 errors, falsely flagging a condition. In policy, the truth of a hypothesis is much less tenable. A position needs to be taken, and uncertainty needs to be specified and managed. Then there's the issue of complexity. I'm going to take you to the top right-hand corner. Ceteris paribus, which is the approach scientists typically take, is not particularly helpful in a policy context, as policies tend to involve a whole systems approach. Understanding complex issues, the kinds of problems policymakers typically deal with, requires diverse and interoperable data from multiple agencies and layers of governance, circumstances that are rarely met. The Erasmus Plus program serves as an example. This program provides great opportunities to study, train, gain experience, and volunteer abroad. A great program, but actually evaluating its impact is not a simple task. Though the policy is set at the European level, the monitoring data are at the national level, and their interoperability is limited. Other times, the difference between policy and science is more of a structural nature. Policy requires extensive synthesis of knowledge, taking you to the bottom left corner. Knowledge synthesis is an activity that academia rarely rewards, unfortunately. Research is too often siloed, and the data are not fair. We've heard that in acronym today. Findable, accessible, interoperable, and reusable. Making data fair and particularly interoperable enables synthesis, which is the cornerstone of science informing policy. And investments are always needed here. And then there's the issue of velocity. That's the bottom right corner. In the social sciences especially, the time frame of research and policy are often out of line. In many cases, policy decisions must be developed rapidly, or at least with a degree of urgency. In science, we tend to be slow. The Survey of Health, Aging, and Retirement Europe, we've just heard about it, SHARE, Alexander Schumacher, and the European Social Survey, we also heard about it, and it was Mark Visser's presentation. They're both excellent data sources, but it takes 2 years from questionnaire design to fieldwork deployment. If you want to know about the impact of a pandemic or a refugee crisis, these are not ideal. But the good news is data collection can be rapidly accelerated. And Alexander Schumacher gave an example. The existing infrastructure means it can be scaled up quickly. In the Netherlands, we have something called the Longitudinal Internet Study of Social Sciences. That's the LIST panel. And during the COVID crisis, we quickly made it open to a wide range of researchers. Within 3 to 4 weeks, the data were there. And so very quickly, and I'm talking April of 2020, we were able to show the differential impact of homeschooling and that it depended on whether your parents had disadvantaged backgrounds or advantaged backgrounds. We were also able to show that homeschooling strengthened a traditional gender division of labor. And we're also even then already able to show that the mental effects, the negative mental effects of the pandemic were much greater for young adults than they were for older people. And everybody thought that the old people would be affected most strongly. In the social sciences, we have both the necessary theory and the data to support synthesis of knowledge for policy. But we're regularly limited by the infrastructural capacity to bring these together. Many of the questions in the social sciences revolve around wicked problems, and social scientists have often sought parsimony in their modeling, which is reinforced by the lack of interoperability in data. This lack of interoperability is partially due to privacy and security concerns, but also due to a lack of adequate research infrastructure to help manage these risks. Through a new generation of social science research infrastructure, it's possible to synthesize diverse, complex, and sensitive data. And complex systems approaches such as fitness landscapes, agent-based modeling, network analysis, and nonlinear modeling are all methods that could be used more effectively if it were possible to securely link a wider range of data and securely analyze them. Such approaches synthesize a huge range of perspectives and priors into a single model with a telling and digestible result for policymakers. During the pandemic, epidemiological models have been deployed for informing COVID policy, but what is lacking is a social science perspective. And one of the reasons is that the social science data cannot be wholly integrated. In the UK, for example, it's not possible to properly link the National Health Service records to wider data systems in the government. And this makes us blind to social and cultural processes which drive health dynamics. Big data and AI hold huge potential. I'm not the first speaker to say that, but they require an evolutionary transformation of research infrastructure. What are the pillars of social science infrastructure that can better serve policy? To start with, data need to be fair. As I've said, findable, accessible, interoperable, and reusable. Second, the 5 safes network, a framework needs to be in place. Safe people, so access is restricted to people who can be trusted with the data. Safe projects, research is limited to projects that are ethical. Safe settings, data are stored in computers with appropriate protection. Safe data, so the data are identified, and safe outputs. Research is reviewed to prevent accidental privacy breaches. Third, following the principles of open science, the data need to follow FACT principles by design: fair, accurate, confidential, and transparent. This being the issue of ensuring reproducibility, not just reusability. Fourth, a crucial point, the infrastructure needs to be driven by research, and the funding must come from the research budget. That's a point that Giorgio Rossi made this morning. This gives the scientists the mandate, which is vital for data integration and curation. Data initiatives, nationally and internationally, rarely adequately service science unless they're led by science. In the Netherlands, the Statistical Office, which has a wealth of social science data, is not mandated to support research. So financing and management of data infrastructure providing access to Statistics Netherlands data must come from the scientific community. The same can be said at the European level. Eurostat and the Joint Research Centre, excellent as they are not substitutes for research infrastructures which facilitate the use of public and private data for social science research. And this is, of course, where ESFRI comes in, and that was a point made by Jean-Éric Paquet this morning. This brings me to Odyssey. A brief overview. Top left, we have 43 member organizations, so we have total community buy-in and some sustainable financing, and we also have national infrastructure funding. Odyssey includes faculties and academy institutes, but also planning bureaus and Statistics Netherlands, so both sides of the community. Take you to the bottom left. Odyssey has 4 key parts. There's the observatory. Its purpose is to sustain scientific data collections like the ERICS, European Social Survey, and SHARE. Then there's the laboratory, facilitating experimental research. Think of mass online experiments. Then there's the data facility, a technical infrastructure to allow linkage of administrative data and scientific data. And the hub, that's all about community management and training. Take you to the top center and right. The infrastructure itself focuses on the integration of scientific and policy knowledge systems, linking surveys and experiments with administrative data in secure, high-performance computational settings. Bottom center and right, fair implementation across government data is key, especially enabling interoperability and data provenance. There are more than 10,000 linkable variables at Statistics Netherlands. That's that pink blob in the slide. And these time series cover decades. Odyssey supports leveraging these for science. A big innovation is the creation of a whole population network. There's 17 million people, 1.8 billion links, and it's dynamic over 10 years. This is a network which describes for every individual in the Netherlands with whom that person works, who the family members are, who the neighborhood members are, with whom this person went to school, who the colleagues are. There's currently a collaboration between the National Public Health Institute, the RIVM, Utrecht University, and my university on analyzing the spread of COVID across this whole population network, allowing for specific key transmission points to be isolated, focusing on questions such as what needs to be shut down first. And the same logic can be applied for economic or social contagions like unemployment or energy-saving behavior. The Netherlands is an advanced case, but it's not unique within Europe. There are similar initiatives in Germany, CASIS, Switzerland, FORCE, the administrative data research in the UK, and of course, France has CUSD. But unfortunately, cross-national infrastructures in the social sciences are highly siloed with respect to administrative data. Share data, for example, can be linked in 6 countries, but the data can only be analyzed in isolation. So you need to use 6 different terminals, and you can't use them simultaneously, and replication across these 6 separate secure environments is hard. Networks like IDAN, the International Data Access Network, try to make this easier, but it's still very laborious for researchers. Eurostat does great work on harmonization and alignment, but not in infrastructure provision. Key policy tools like Euromod, which is a tax benefit microsimulation model run by the Joint Research Center, fail scientific standards of transparency and open science. What is needed is infrastructure that's shared infrastructure, where strengths of the Joint Research Center, the European Commission, Eurostat, and the cross-national surveys can be combined with those of the scientific community. In short, what I envision is a European-level integration of existing national infrastructures that creates a large social science collider, where administrative data and scientific data from across the Union can be securely combined and made fair. These systems must enable full use of the e-infrastructures that exist in Europe so that we can enable computational social science for policy making at the European level. Currently, the substantive silos in the SSH-ERICS prevent this, leading to an underutilization of e-infra by social science and a limited policy impact of the social sciences in Europe. The urgent and complex policy issues are out there. The social sciences are more than ready to address them, but to properly do so, the data need to be brought together. Thank you.
Thank you. We have time for one burning question. Should be burning? No? Well, I think they're all looking for the coffee break. Oh, you have one here? Okay, sure.
I'll try it as a natural scientist who is suspicious about political processes in transition when they go unstable. And I wonder if you have as nice as it sounds that Europe has everywhere the same thing, is how much that makes the system very unstable when you have all of a sudden, by chance, a government which you don't want to have. Then I would think, as much as you like the equalness, otherwise, at that point you pay for it badly.
Yes. This is a very pertinent question. And I'm thinking of what happened in your country with the Environmental Protection Agency. So yes, the political clout has to be there, but it's also crucial that our research infrastructures are run by the scientists. That's what I've emphasized. And it's up to us to maintain secure systems. You wouldn't want to know, like this secure supercomputer we have, how often we do all kinds of pen tests to make sure that it's secure. We're talking about sensitive data, but they are very important data, but also by analyzing them, by showing how we can work together, but also by showing the population what can be done with data that they have provided with us. We also attempt to create trust in science and to make them also more a participant in science.
I know that the wish of the scientists or the philosophers to be in charge is as old as Plato. But the thing is, in Germany anyhow, it must have been about a year into the pandemic, the voices got louder and louder. We are here in a dictatorship of the virologists, and they have no political mandate to tell us what to do.
Your point is well taken, but I'm not exactly sure what that would mean for enabling research on the virus, as the previous speaker said. At least once we have those data, we can also examine the backgrounds of people and better explain what the effects are of vaccination or of not vaccination. One of the unfortunate circumstances right now is that the vaccination data are in the biomedical sphere, and they are not linked with information on factors like gender, factors like age, where a person lives, what the educational attainment is, and that enables us to much better examine those who have provided their vaccination data or who have been vaccinated. So, and still, perhaps I'm too naively optimistic, but I do think that by analyzing these kinds of data in a very careful way and communicating them in a thoughtful way, and I think that's where the communication sciences can help us in how we bring measures, we might create more trust, a better trustworthy relationship with our population. But thank you for your questions.
Thank you, Pearl. We have to move on. So our last speaker for this session is a young researcher, Rami Al-Shambari. Please come on. Who works at the University of Exeter. His work is related to the European Plate Observing System, the EPOS infrastructure. And his talk will be about modeling volcano deformation, a perspective on recent developments and future challenges. It's all yours.
Hello, thank you for the introduction. So, again, I'm Rami Al-Shambari. I'm a PhD student at the University of Exeter. So, in today's talk, I'll be talking about the science of volcanoes and volcanology in general, and I will focus more about my field of study about volcanogeodesy. And I'll briefly go through my involvement with EPOS project. So, volcanism is a unique feature of our planet. So I will start with a few facts and a few figures about volcanoes on our planet Earth. There are 551 historically active volcanoes, and as we can see in this map, that most of these volcanoes are distributed along the plate boundaries. So these volcanoes are a result of interaction between two plates, and we have around 50 eruptions per year. We don't hear about most of them because most of them happen in a remote island, so it doesn't affect a lot of people and doesn't pay the media attention. So nowadays, there are around 300 million people within the danger range of an active volcano. So 1 person in 20 people, they live within that range. So although the volcano activity is not increasing in our planet, but the volcanic risk is increasing because the global vulnerability is increasing. So the population growth around active volcanoes is growing, and our reliance on the infrastructure is increasing. So volcanoes produces a wide range of hazards with enormous risk. And here I'm showing 6 kinds of hazards, and I believe there are more. So the first one is the tephra, the ashfall, and the next is the lava flow. And as we saw 3 months ago, how the lava flow caused a significant destruction to the infrastructure in La Palma. And then we have the lahar and the volcanic gas, which can cause health difficulties. And then we have the pyroclastic flow, which is avalanches of hot rock and dust that travel very fast, and it's very dangerous. And finally, we have the landslide. And if the landslide is significant, it can cause tsunami, like what happened in Krakatau in Indonesia in 2018, which left more than 400 deaths. So in order to mitigate the volcanic hazard, a continuous scientific investigation is required for these volcanoes and a continuous monitoring of these volcanoes. So this brings me to the term volcanology, which is the science of volcano, which concerns the geological and the geophysical and the geochemical phenomena associated with volcanism. So as we can see from this definition, the volcanism is a multidisciplinary field. So every volcanologist tries to answer 2 key questions. So the first question, we want to know what the time, the location, and magnitude of a future eruption. And next, we want to know the duration and style of eruption. Either it's an explosive or effusive eruption once initiated. So this brings me to our research focus. We want to know what's under a volcano, the depth and the shape of the magmatic reservoir where the magma is stored, and the composition of that magma and the process involved. So we know that the magma is stored deep in the earth, so we can't directly observe these magma. So we use different techniques and different methods to track a volcano, but in this talk, I'll just focus about my field of study, which is a volcano deformation. We can observe these deformations using different techniques, ground-based techniques like the tilt meter and the GPS, or a satellite-based technique like the remote sensing and the INSAR. So let's start with a quick tutorial about the volcano geodesy. So what causes these deformations and what the forces behind these deformations. So when the magma rises from the upper mantle because it's buoyant and accumulates into a crustal reservoir, the internal pressure of this reservoir will increase and we will have the inflation. When the pressure within the reservoir reaches a threshold value, so the reservoir can sustain more pressure, the magma will exit this reservoir either through a conduit or a dike, and the internal pressure of this reservoir will decrease and we will have the deflation. So in a few words, the inflation is associated with a new magma accumulation and the deflation is associated with a withdrawal events or eruption. So the figure at the bottom, I'm showing the tilt meter recording from Kraftline, Iceland. So as we can see, the inflation-deflation cycle, up is inflation and down is deflation. And some of these deflation are associated with an eruption, and some not, because the magma can rest into another level within the crust. So here I'm showing 2 different studies of the same volcano, which is a Sierra Negra volcano. In the first figure, I'm showing the INSAR imaging of a volcano. So INSAR basically takes 2 different images of the volcano surface in 2 different times and measure the change within the phase. And by knowing the change in the phase, we can see how much the earth deformed. So we can measure the deformation by counting the number of fringes in each of these 3 figures in the first one. And in the second figure, I'm showing a combination between a ground-based instrument and a space-based instrument. So as we can see, so from 1992 to 1998, they used the INSAR, and then a campaign GBS and continuous GBS after that. So INSAR gives us a global view of the deforming volcanoes, while the continuous GBS gives us a high resolution, temporal resolution deformation. So these deformation data typically interpret using what we call the Mugi model, which is a widely used model. So this model considers a pressure source point embedded in an elastic half space. So this model is widely used because it has such a simple formula for the vertical and the radial component of the deformation. So this model has a limitation. So first of all, it has no predictive power because it's a kinematic model. And secondly, this model considers the magmatic system to be a highly melt fraction magmatic system. And this contradicts the seismic tomographic images of volcano, the studies. So as you can see from this cartoon, which is based on a seismic tomographic imaging of Yellowstone caldera, which is in the Cascades, would suggest that the upper part of the magmatic system contains just a 9% melt, while the lower part just contains a 2% melt. So the system is not melt-dominated. It's not a single melt-dominated chamber within the elastic half space. And here another different studies I'm showing here. So as you can see, the maximum reported melt fraction is from Altiplano Puna in the Andes, which has an average of 40%. Moreover, the physical models of volcanic process, the thermal and mechanical models, suggest that it's not easy for the system to be melt-dominated for a long period of time. And instead we have the new paradigm, which is the transcrustal magmatic system, that suggests that the system is a crystal-dominated system. So, as I mentioned before, when the magma cools, the crystal fraction within the magma will increase, and when it reaches a critical point of crystallinity, the crystal will interact with each other and form a framework of crystal mush. We call it crystal mush, which is a highly crystalline magma. So in our study, we consider that the mush behave in poroelasticity. The easiest way to demonstrate the poroelasticity by a sponge. So when we inject water into the sponge, the pore pressure in that sponge will increase and it will inflate. And when we take that water out of that sponge, the pore pressure will decrease and it will shrink. So we consider the mush to behave in a poroelastic behavior. So I'm showing some results. So the two figures at the top, they show the deformation evolution with time. We apply an injection source at the bottom of the reservoir. So in the case of melt-dominated, the system will react instantaneously. And in the case of melt fraction, the system will continue to evolve. So finally, since that conference is about celebrating 20 years of infrastructure sharing, I just want to point out that some of my study was funded by the EuroVolc Transnational Axis, which is a European project that gives volcanologists a unique opportunity to generate cutting-edge research and implement technological innovation, and most importantly, build a collaboration with other institutions. So this project aims to build a new research collaboration among 13 European infrastructures, with over 30 facilities. So the EuroVolc, through EPOS, gives us the opportunity to visit Geoscience Barcelona and have on-site access to the computational infrastructure there and run our models. And finally, I want to thank my supervisor, who's also James Hickey, who's also involved in that project. Thank you for listening, and I'll be very happy to have your questions. Thank you.